Good morning, I'm Yayu Hu. Today I'm going to talk about the role of glial cell under excitotoxic condition. The title of this paper is Reactivity of Microglia and Astrocytes after Excitotoxic Injury Induced by Kinic Acid in the Rat Spinal Cord. We all know glia, also called glial cells, are non-neuronal cells in the central nervous system. They maintain homeostasis, form myelin, and provide support and protection. When central nervous system damage, it will cause glial-active coaguliosis. For example, mature astrocytes will become reactive cells under the influence of damage-associated signals. Microglia may change their morphology and secrete pro and anti-inflammatory materials. Excitotoxicity is a pathological process which neurons are damaged and killed by the overactivations of receptors for the glutamate. It occurs in several neurological disorders. Kinic acid is an analog of glutamate and is widely used as an excitotoxic agent to lesion spinal cord networks. This can provide an interesting model to learn basic mechanisms of spinal cord injury. Thanks to the previous studies, we already know that glial cells can be activated in different excitotoxic-induced neurological disorders. Although gliosis was mentioned, no counting of those cells was performed, nor detailed description of their morphology was reported. Therefore, in this study, the author tried to examine the morphological reaction of astrocytes and microglia after the injection of Kinic acid in a rat spinal cord. In this experiment, the author used male rats to study the role of glial cell after excitotoxic injury. Rats are divided into three groups: control, vehicle, and Kinic acid group. After injection, they collected the spinal cord tissues in the post-injection day 1, day 2, day 3, and day 7. Then used these tissues for his pathological analysis, immunohistochemistry, apoptosis, and neuronal degeneration determinations. First, the author found that the Kinic acid injection caused extensive neuronal loss and glial cell active. Further focus on the glial cell morphology after Kinic acid injection. Figure C represents the morphology of IBA1 positive cells. Figure E represents the morphology of GFAP positive cells, and Figure D and F are higher magnification of Figure C and E. Next, the author examined the ipsilateral and contralateral sites. In Figure 2a, the Kinic acid group show higher numbers of IBA1 positive cells at the post-injection day 3 at both sides. In Figure 2b, significant differences percentage of IBA1 positive cells between vehicle and Kinic acid group were found at both sides. And the Figure 2c1 to c5 indicate the different microglial cell phenotypes. Then it's time to examine the GFAP positive cell numbers. The significant differences were found at post-injection day 3 at both sides. The global branch lengths of GFAP positive cells were measured from more complex branching at post-injection day 3 at the ipsilateral side. Then, the author tried to detect proliferating glial cells. It revealed that IBA1 positive cells and GFAP positive cells colocalizing pCNA in both sides. Finally, the author identified degenerative and apoptotic cells and found both their positive cells were found in Kinic acid group. Okay, there are two take-home messages. First, a general reduction in the number of microglia by post-injection day 7 as compared to day 3, and particularly a reduction in the percentage of active microglia phenotypes, which may be related to a neuroprotective function. Second, This study provides a novel information about phenotypical changes occurring on glial cells after the excitotoxic effect. Understanding the involvement of glial cells in different kinase acid-induced models may help to find specific targets for therapeutic strategies. Thank you.